Thank you very much, Neta, for the extensive and interesting presentation. I think they are representative from other pharma companies. I saw Excel Pharma is moving also from generic to innovative, and we hope to communicate with the entire pharmaceutical industry in Israel. The next speaker is Batsheva Kerem from the Hebrew University, a pioneer in the field of rare disease with important contribution to the discovery of the gene for cystic fibrosis. Was awarded with the Met Prize, and we're looking very much for your talk.
כתוב עליה, כתוב קרם. הראשונה. So, good afternoon. I would like to congratulate Miguel for establishment of this encouraging center, for organizing the day, and for inviting me to present some of our results. What I would like to do today in this short lecture is providing an overview of the research in cystic fibrosis aimed at identification of drugs for CF. So I will start with this, and then in the second half of the lecture, I will present some of our efforts in this field. So we are talking about cystic fibrosis. Cystic fibrosis was mentioned already in the conference, but I would like to give some introduction so we will understand the challenges and the success. So CF is a life-threatening genetic disease that primarily affects the lungs and also the gastrointestinal system, as well as the sweat glands, and in males, in men, the reproductive system. It's a relatively common among Caucasians, and the frequency is 1 in 2,500 live births. So it's not very, very rare, but it's definitely not a common disease. There are registered 70,000 patients in the US and in Europe. There are many more in South America, in Eastern Europe, but at least we do have clinical and, in many cases, genetic data on at least 70,000. The gene was found exactly 27 years ago. It will be tomorrow, September 9, 1989. So it's 27 years. And the gene that was found encodes a chloride channel. And here we see a model of the channel. It's a very unique gene, a long gene and a long protein. And this chloride channel is functioning and expressing in epithelial cells. And later on, we found, as the community, that it has another very important role, not only as a channel, but also as a regulator of another epithelial, respiratory epithelial channel, which is a sodium channel called ENaC. There are over 1,000 mutations in the Cftr gene found among patients worldwide. And the mutations happen along the entire gene. There is no hotspot. I would like to mention that in our patients in Israel, both Jewish and Arabs, the number of mutations is very small relative to the 2,000 and includes less than 100 mutations. The mutations were a long time ago divided into classes according to the effect of the mutation on the protein function. And you will see how important this is for identification and development of drugs for CF. So mutations in general are divided into 5 classes. Class 1 generates defective production of a protein, and this includes also nonsense mutations, as the nonsense mutation due to nonsense-mediated messenger RNA results in almost no production of the protein. The second group, which includes also the major mutation worldwide, by the way, this mutation is not a major mutation in our population. So class 2 mutations, the mutations cause defective processing of the protein. And most of the protein is not reaching the apical membrane of the epithelial cells. Class 3, which is very important for our discussion today, has defective regulation, and I will show this in more details. Class 4 includes mutation with defective conductance, and class 5, this is the class of mutations that we work in my own lab, is leading to reduced levels of the normal protein due to splicing mutations. And the approach, as we heard in the keynote lecture this morning, we do have the hope that gene therapy will indeed be developed and be successful, but at the moment, for most of the diseases, this is not available due to many, many reasons. So in CF, a mutation-specific therapy approach is the main approach, trying to develop therapy according to the classes of the mutation. And actually, the North American CF Foundation, which is a very important organization and a driving force of research in CF, are declaring that they will reach each and every patient, even with very rare mutations within the Cftr gene. So the first story that I want to tell you is a very successful story in developing a drug for class 3 mutations, defective regulation. So the protein is synthesized, it is correctly processed, it is correctly inserted into the membrane. However, the chloride channel activity is strongly reduced due to what we call gating defect, and I'll show you in a moment. And the mutation that the drug was developed for is called G551D. The name is not important. And there are, in the registries, 850 patients. And the drug was developed for these patients. So we can see here a record of the chloride efflux and chloride channel activity in a cell line from a patient carrying the G551D mutation. So we can see a very limited opening, not very high, and for a very, very short time. So we can see here another very time-restricted opening of the gating, of the channel. Here it's a little bit better, but it is very, very poor. A library of small molecules was screened, and one molecule, initially called VX770, was identified. And as we can see here, we can see the effect of this VX770 on the opening probability of the channel. And this was developed as a drug, and I have not enough words to describe the effect of the drug and the feeling that going from a gene to a drug. So we can see here the results of the clinical trial, phase 3, that led to the approval of the drug. We can see here measurement of lung function. As I indicated in the beginning, this is a lung disease, and the major morbidity and mortality cause for CF is a chronic progressive lung disease. Patient needs a lung transplantation or they lose their life. So we can see that along a year, typical for a CF patient, about 2% of the lung function is lost. We can see here that patients treated with VX770, we can see that in 2 weeks, there was an increase in lung function of about 10%. This is unheard of in CF. We initially were thinking that we aim for stabilization of the decline of lung function in CF patients. And What we saw here is in 2 weeks an high improvement which was stable along the treatment. So this was really, really a milestone in cystic fibrosis. And we can see here that the sweat chloride was normalized in 2 weeks. We actually know exactly if patients are taking the drug or not, because if they don't take the drug, in few days, we do see the aberrant sweat chloride levels. So they can tell us that they are taking the drug, but if their sweat chloride is up again, there's no way they are taking the drug properly. So the effect on the sweat chloride is dramatic as well. And I won't go to too many clinical details, but they gained weight, which is a big issue in CF, and height, etc. And this was 20 years after cloning the Cftr gene. So 7 years ago, this drug was approved. So it's a milestone, and this is the name of the drug that we are very happy about. In Israel, we do not have any patient with G551D mutation. However, following the approval of the drug, other mutations from class 3, from the same class, showed response to the drug. So the indication, the approval, was expanded to additional 9 mutations, so it's 10 mutations altogether. And we have in Israel now 16 patients that can be treated because they have class 3 gating mutations. Just to talk about price, since this was already raised, the cost of the drug is unbelievably high. It's $300,000 per patient per year. I will repeat the number, $300,000 per patient per year. But the drug is doing fantastic for many patients, not all of them. And as my husband, who is working in the field, said, we had last year 3 weddings of patients in a very, very good condition. So they even could dance. Usually they cannot breathe, they cannot exercise, they cannot sing. And it was really, I was there in these weddings, it was really, really moving. So this is a very successful story for class 3. Class 2, many patients, most of our patients, not in our population, but in the world, carry at least one allele of a mutation. For those who know the field, is the delta F508, belonging to this class. So the mutation impairs the folding of the newly synthesized CFTR in the ER, and as a result, there is a dramatic reduction in the transport of nascent CFTR proteins from the ER to the Golgi and to the membrane. This leads to loss of function at the cell surface. And there is accumulation of the protein, of the CFTR protein in the ER, subjected to degradation. So I would like to show here a normal cell, a normal epithelial, respiratory epithelial cells. We can see here the nucleus, and we can see here the position of the CFTR on the membrane. And here is a cell from a patient homozygote for the delta F508 mutation, and we can see no apical appearance. It's all in the ER, and the level is low. And it turns out that aside from the trafficking defect, so protein is degraded and it doesn't make it to the membrane, this mutant has impaired chloride channel activity characterized by reduced open probability. So the very, very low amount of CFTR with this mutation, which make it to the membrane, is suffering actually from the same defect as the G551D gating mutation. So the therapeutic approach for these patients requires both a corrector, that's the way we call it, which is molecule aimed to correct the transport defect, and a potentiator which will increase the open probability of the channel. And this happened, and in July 2015, 2 years ago, a drug was approved by the FDA. It is called Orkambi. It's a combination of drug between the first drug that we discussed and a corrector. However, its effect is quite limited. It is stabilizing the decline in all the parameters, so there is stabilization. And in lung function, for example, probably we do have on average a 2% improvement in lung function. So there is more, or the drug is used also for our patients. And the price is close to $500,000 per patient per year. I am embarrassed to say these numbers, although I have no responsibility for them. Okay, so now I would like to share with you some of the experiments and efforts that we do in my lab, concentrating on class 5 mutation. Before that, I may say that there is a huge effort to develop a drug for class 1 mutation with stop mutations. We are involved in this as well, and probably class 4 will be the most difficult for developing drugs. So, I said that class 5 mutations include splicing mutation. So I would like to give a bit of a background. In the Cftr gene, there are 27 exons that are all required for the normal function of the gene. Differently from other genes, or most of the genes, we do not have any functional alternatively spliced isoform that we know of in the Cftr gene, messenger RNA, or protein. We do have only one isoform that should include all the 27 exons. And about 10 to 15% of the mutations are splicing mutations, mutations that affect the splicing. So we all know what splicing is, just a few words of introduction. We know that splicing is determined in cis by sequences, and we do have the donor, the acceptor, the polypyrimidine tract, the A branch. But in later years, it became apparent, and we are all convinced that there are other sequences that we define as enhancers of splicing and silencers of splicing that appear both in introns and both in exons and in introns. So these are the cis sequences. And as we all know, proteins have to recognize these sequences and operate in the splicing machinery. So here are the splicing factors, and many of them are known. So the aim of the project in my lab is to modulate the level of correctly spliced CFTR transcript to enable higher levels of functional CFTR channel and aiming to improve the CF disease severity and the patient's quality of life. And I would like to share with you results on one mutation that we are focusing. The name of the mutation is 3849, since this is the number of the last nucleotide of exon 22. So 10 kb downstream of the last nucleotide of exon 22, deep into the intron, there is a C to T change, but it is not an innocent change, as this C to T mutation generates a donor site, and since we all have an AG, 84 base pairs upstream, and also quite a strong polypyrimidine tract, as a result of this C to T mutation, now these 84 base pair can be recognized as an exon, as a cryptic exon. And in frame, there is a stop codon, okay? So this inclusion of the cryptic exon leads to a nonsense mutation and no protein. But from the first patient who was discovered with this mutation, it was a patient from Pakistan, homozygous for this mutation, it was clear that despite the mutation, some correct splicing also occurs from this specific allele. So this mutation can lead to both aberrantly spliced transcript as well as correct spliced. And there are over 1,100 patients in US and Europe, including Israel, carrying this mutation. The mutation has a variable disease phenotype from very severe classical disease progression and severity to patients with a more milder disease. And what we did several years ago, although on a small number of patients, we analyzed the level of correct and aberrant splicing and looked at the lung function of these patients. And I think that you can see here a very clear correlation between the level of correct splicing. As the level increases, the lung function is increasing. So this led us to think that maybe if there is a variability in the level of splicing, of aberrant splicing and correct splicing, maybe we can do something about it and modulate in order to improve. So we studied the effect. So we started by studying the effect of overexpression of splicing factors on the CFTR splicing pattern and protein function. As a proof of concept, can we modulate the splicing pattern, and will this have an effect on the CFTR function? So we overexpressed several splicing factors from different splicing families. All of them are quite essential in splicing, in general splicing. And what we can see here, that in one case, and we measure here the level of aberrant transcript. So in one case, the splicing factor increased the aberrant splicing. This is not what we aimed for. In other cases, there was no change. But luckily, 3 of the splicing factors significantly reduced the level of aberrant splicing. And when we measured the chloride efflux following the overexpression of these 3 factors, we found that 2 of them significantly increased the chloride efflux after the treatment. If we have time at the end, we can discuss why the 3rd one did not have this effect. So the conclusion from the set of experiment was that reducing the level of aberrantly spliced transcript containing the aberrant exon can restore the CFTR function. And I think that at this point I would like to mention that we know now that the level of normal messenger RNA and normal CFTR protein that is required for normal function is quite low. It's probably below 15%. We all know that 50% is absolutely fine, right? All the heterozygotes, they are healthy, no problem. And the question was, how low can we go down from the 50%? And what we understand, and my lab contributed to this analysis, we are, I think, convinced now that it should be around 50%, if not lower than that. So at the end, I would like to show you that we are now trying to develop a more targeted approach that will not affect all exons in all genes, as splicing factors are expected to. And antisense oligonucleotide are small synthetic, chemically modified nucleic acids able to bind specific to intronic or exonic sequences, and in our discussion to intronic or exonic splicing sequences. And if the aim of the antisense oligonucleotide is to bind to the pre-messenger RNA and mask the sequence. So proteins will not be able so efficiently to bind to these sequences. So masking splice junctions or splicing enhancers, this is expected to reduce exon recognition, as we would like to see in the 3849 mutation with the 84 base pair cryptic exon inclusion. And the reverse is, of course, true, that if we will mask splicing silencer, we will promote exon inclusion. And there are many mutations in which we want to promote exon inclusion. And there is an extensive effort to develop antisense oligonucleotide approach for other diseases. So we are using a specific chemistry, a specific modification of the antisense oligonucleotide, which is fully 2O methylated phosphotioate nucleotides, and our antisense oligonucleotide are between 20 and 25 nucleosides. And we designed several antisense oligonucleotides, 2 of them to mask the junction, and 2 of them within this cryptic exon. And the first result I want to show is from a respiratory epithelial cell of one of our patients. The cells are heterozygote for the splicing mutation. And you can see that using all these 4 nucleotides, we got a very, very strong reduction in the level of aberrant splicing. And because there is a stop mutation, and we expect nonsense-mediated messenger RNA decay, if we can avoid the generation of transcripts that are subjected to NMD, we should see an increase in the total level of the messenger RNA. And this is what we see here. And we do see it in human bronchial epithelial cells, primary cells. The first result was in a cell line. This is in human bronchial epithelial cells. You can see the same strong effect. Can we affect the function? The answer is yes. So we can see here the effect of the antisense oligonucleotide on the CFTR function in one cell, in other cells. Here it's without transfection. And hopefully the AOs will have, or they have the potential, hopefully they will make it, to correct the CFTR function in patients carrying the endogenous allele with the 3849 mutation. And the work is done in my lab by these people who are very talented and devoted, and actually started with a former PhD student, Malka Nissimov. We have a collaboration with Steve Rowe from the University of Alabama, and here is the funding. Thank you.